Good morning. A very good morning and welcome to Unacademy. So, guys, as per our planning, we today, the Saturday, we held for the revision. Okay, so today we are going to revise the complete chapter of cell division because it is a one of the very important chapter. Okay, so it is necessary to understood it more and more. So we can attempt the most of the question which is coming in the examination on this topic. Okay, so this topic is not directly asked in the examination, but if you see the spermatogenesis and the oogenesis, it is completely depending upon the meiosis part of this topic. And the second thing, the mitosis is always help to understand the many other thing inside of the body. Okay, so it is very necessary to understand the cell division and the stages involved in the cell division. Okay, so let's start with a cell division, but directly before starting the lecture it is necessary to know about few things. Okay, so the first of all, this is the Crack Maharashtra CET YouTube channel. Okay, so this channel is for the MHT CET 2021 examination, and if you see the name of a batch, that is the Target Top 100 Ranks MHT CET 2021 batch. Okay, so this channel will give you the detailed knowledge and the full information about the MST CET, so you can boost your score. So this is completely for the to boosting your score. And if you see the timing as per your PCB group or PCM group or PCMB, okay, so whatever it is, so the timing is in front of you. Mathematics 10 a.m. and 8 p.m. So daily two lectures are there with chemistry 1 p.m. and 9 p.m. Physics 9 a.m. and 6 p.m. and biology. This is the 11 a.m. The first lecture of biology and the second lecture of biology at the 7 p.m. So if you see the syllabus, the morning lecture is held for the syllabus of 11th when 11th completed. So we will start 12 also at this point, and the evening lecture is held for the 12th syllabus. Okay, so there is the topic, the reproduction in an animal is going on. So you, if you want to attain the 12th syllabus, so please be on time at 7 p.m. Okay, so before starting the lecture, it's necessary to know about me, guys. So this is Abhijit Madkar. I have more than 6 years of experience in the field of biology and also crack net CSIR from the life science. Okay, so uh, this is, the India's largest platform where we are now. Okay, so the Unacademy is India's largest platform. If you want to enjoy more education with the Unacademy, so it is necessary to install this app in your mobile. This app is available on the App Store. So whatever type of mobile you are using, this app is available there for you guys. Okay, so let's say the Telegram app. This is the channel, actually Telegram channel, which is help you in the daily updates and the notes plus MCQs. Okay, so if you do not have join, please join this channel. Okay, so very good morning, guys. And this is the subscription, the facilities for you guys. So this is the very good facility. If you take a subscription, so you will have the few options open, like a offline lecture, so you can download the lecture and you can study as per your time. The second thing, you can interact with your favorite educator. The third thing, the dedicated doubt solving session. And the fourth very important thing is the MCQs practice. So there are lots of MCQs paper available. You can practice it and you can analyze it after the examination. So you will have the detailed report after the examination, that is the which type of question you make correct, which type of question you make incorrect. This type of information is available after the examination, so that the doctor of MCQs will definitely help you to boost your score. Okay, so these are the few facilities I told you, but if you see in the description of a video, there is a lot of facility which is comes after the subscription. So if you want to take a subscription, guys, take a subscription of 12 month or the 24 month. So for the 12 month, if you see the fee, it's a 14,000, and for 24 months it's a 21,000. I will recommend you, if you are in a 12, so take a subscription of 12 month. If you are in a 11, take a subscription of 24 months. So it will help you till the examination. Okay, so that is CET. Till the CET examination, it will help you. So take a subscription. The longer, if you see the 12 month and 6 month, there is a difference of a 6 month, but the fee, if you see, only 2000 and 2000 few hundred is a difference. So only 2 to 3,000 will increase your complete 6 months. So take a subscription of a longer one will definitely help you economically. Okay, so thank you very much, guys. And at the time of a subscription, it is very necessary to take this Abhijit, capital Abhijit, as a subscription code. It will again reduce your fee. Okay, it is for the 10% off, guys. So this name will definitely reduce your fee. Okay, so let's start with the revision of a cell division. So here is cell division. You can see in the diagram. Okay, so the one cell which is going to be divided by this plane and this plane also. So the next one is the plane is here. Okay, so this is nothing but the embryo like a structure which is going to divide again and again and again and again. Okay, so embryo is always start division one by one, and it will not going to stop, I mean not going to stop its division uh, before the birth, and after birth it is also the going on. Okay, so but if you see the embryo, it is a very fast in a division for few days, first 1 to 2 week, because if you see for first few days the size of embryo is not going to increase, the size of ova is fixed, but only the divisions are going to be very fast. Okay, so like this. So this division increases the number of a cell with respect to the time, and if you see the complete, the finally cells enters into the growth phase, and then they start increase in size. Otherwise, the first few days, so size of embryo is not going to increase. So this type of the divisions one after one is started and allowed only in a embryo. So not other part of your body is going to be divide again and again one by one division. Okay, so it will take definitely the time between the two division, because it is necessary to increase the volume of a cell before the division. Okay, that is the growth, you can say the growth of a cell. Okay, so in embryo it is not required, because there is a special arrangement and the embryo is in a uterine tube. So there is again the a different type of environment which help it to divide again and again. Okay, so we will discuss it into the evening lecture. Okay, so that there is we are going to talk about the revision of embryology, that is the developmental biology. Okay, so please be on a time there. तिथे तरी वेळेवर या guys, कारण तो जो टॉपिक आहे, तर तो टॉपिक तुमचा 12वीचा टॉपिक आहे आणि अतिशय महत्त्वाचा आहे. अतिशय महत्त्वाचा आहे हे तर तुम्हाला माहितच आहे, एम्ब्रियोलॉजी. ओके, आणि तो समजणं, जर तो तिथे का महत्त्वाचा आहे, जर तो तिथे नाही समजला ना, परंतु दुसरीकडे नाहीच तुम्हाला सिलेबस मध्ये, डायरेक्ट तुम्हाला पेपर मध्ये येईल तो, त्याचं म्हणजे कुठं नाव पण काढलेलं नाही तुमच्या सिलेबस मध्ये. त्यामुळे गरजेचं आहे की तुम्ही तिथे वेळेवर असलं पाहिजे. ठीक आहे, कोणाला काही शंका असतील तर विचारत चला. So this is the phases, and you can say this is the actually the path of the cell division. Okay, so the cell division always start from the G1 phase. So this is a G1 phase. Cell always enter into the G1 phase for the division. Okay, so cell is going to enter into the G1 phase. So after G1 phase, it will enter into the S phase. After S phase, it will enter into the G2 phase, and after G2 phase, cell will enter into the M phase, where the nucleus is going to be divided into the two, and the two cells are synthesized. One is here and second is here. Okay, so the after two cells, the cell either enter into the cell division, or the second chance to had a cell is G0 phase. Okay, so G0 phase is called as resting phase. याला resting phase म्हणतात. So in this phase, cell can rest for the limited time, or you can say the longer time, from the path of a cell division. Okay, so for example, let's say this is the highway. Okay, here जो cell division दिला ना G1, S, G2 आणि M, हा highway आहे for example. तर हायवे वरती आपल्याला थांबता येतं का मित्रांनो? नाही थांबता येत. आपण कुठेतरी जागा पाहतो, बे म्हणतो आपण त्याला, की साईडला कुठेतरी जागा पाहतो जिथे आपण थांबू शकतो आपली गाडी. मग ती जी जागा आहे त्याला रेस्टिंग फेज म्हणा थोडा वेळ. ओके, मग ज्या वेळेस सेलला थोड्या वेळासाठी थांबायचं असतं, तर त्यावेळेस सेल जी नॉट फेज मध्ये येते. सो इफ यू सी द nerve cells, they are arrested in a G naught phase. So that's why not going to enter into the cell division. So cell division is a myth for the nerve cell after that particular age of maturation. So they will not going to enter into the cell division again, because they are arrested in the G naught. Okay, so the G naught phase, if you see, the G naught phase is a very important thing. So first of all, why cell enter into the G naught phase? So the very easiest answer for that is the differentiation, guys. Okay, so the easy answer for that is the differentiation. So you know that what is differentiation, what is redifferentiation, what is de-differentiation. All the things we already explained in the last lecture. So this is the reason of a differentiation. Okay, so differentiation means maturation. Maturation means taking a particular function. Okay, so the specialization of cell for particular function. Okay, so this is what we call it, the maturation or the differentiation. Okay, so the G naught or G0. So this is the resting phase where cell can stand for a longer out of the cell division. If cell is inside of a cell division, it will not going to stop for a longer. So the cell, any cell which is inside of the cell division, that is the G1, S, or G2 or M, it will continue its cycle of a division, and it will increase the number of. Okay, so if it happens, so it will result into the cancer cell, guys. So the cancer cell never stop into the G naught phase. The cancer cell will not going to enter into the G naught phase. It will continue its pathway like this. Okay, and not going to stop anywhere. So it will just move like the G1 to S, G2 to M, and again M to cytokinesis, and cytokinesis to again G1. So the continue division will happen inside of the uh, cancer cell. Okay, so we will have the detailed knowledge of a cancer in the special lecture of cancer, so you don't worry about that. I will definitely yeah, I will definitely take that lecture in the vacation of Diwali. Okay, so the Diwali vacation is we will spend with the different type of diseases. Okay, so that is the chapter we can discuss in the division offline. Okay, so that's why I'm selected it as the chapter in the Diwali vacation. Okay, so the lecture will be very short and the interesting also. Okay, that's why the only half-hour lecture I will take into the that 2-3 days. Okay, so don't worry, I'm not going to take your much more time in the vacation of Diwali. Okay, so let's see, guys. If you see the time in the phases, okay, so the largest phase or the longest phase is a G1 phase. Okay, so this is the longest one. Okay, and this M1 sorry, M is the smallest one, or the short phase. एकदम छोटा है, or the short phase. Okay, so this is the G1, yes, G2 and M phase. Okay, so what they actually does. Okay, so let's see, guys, what is meant by G1. So G stands for the growth. So S stands for the synthesis. So this G is also for the growth. Okay, and M for the mitosis. Okay, so let's see here for mitosis in the meiosis, it's for meiosis. Okay, so here let's see what happened. So this is your G0 phase, terminally differentiated cell withdrawal from the cell cycle for the indefinite period. Okay, so the cell is coming out of the cycle, so it will not going to enter into the cycle and divide. Okay, so the cell, when cell want to again enter into the cell cycle, so it will definitely enter into the G1 phase again. Okay, so the G1 is very important for the cell to uh, move out of the cell division and coming in inside of the cell division. Okay, so keep in mind that this thing. So in a G1 phase, it's the synthesis of protein and RNA is happen in a G1 phase. If you see, if you understand the G1 phase very well, so guys, let's say, so here is your S phase after G1. Okay, so the G1 after G1 cell is going to enter into the S phase. So the G1 is nothing but the preparation. Okay, so it is nothing but the preparation of cell for S phase. Okay, and this S is the phase where DNA is a synthesis. Hello guys, is it audible? Is it clear guys? Now is it clear? Okay. Chalo, sorry for inconvenience, guys. Actually, uh, yeah, it's okay now. Okay, now okay. Okay, thank you very much. Chalo, let's start. So, we were talking about the S phase. Okay, so actually the G1 where the proteins and RNA are synthesized, so these proteins and RNAs are very necessary for the S phase. So you can say this G1 phase is nothing but the preparation for the S phase, and this G2 phase is nothing but the preparation for a M phase. Okay, so the S phase is actually the phase where DNA is replicated, that is the making of a copy of a DNA. Okay, so the making of copy of DNA will they ensure that the cell of a 46 chromosome will definitely divide into the two cell of a 46 chromosome. Okay, and this is the logic behind the S phase. Actually the S phase, if you see the second largest phase, it will take the 6 to 8 hour. Okay, so the preparation for this phase will take the maximum time of a cell division, that is 6 to 12 hour. Okay, so in this phase actually the everything is checked. So what is the required for the S phase? So the making of a copy of a DNA required, first thing is ATGC. Okay, so these are the nucleic acid, which is very important for the making of a DNA. Second thing is accessory proteins are required, energy is required, and the most important thing, enzyme is required, that is the pol 1. Okay, so the pol enzyme is very important. Pol 1 means not pol 1 2 3 4. Pol means the polymerase. Okay, so I want to say the polymerase enzyme. So this polymerase enzyme is very important to make the copy of a DNA, or you can say it's responsible for the polymerization of a nucleic acid. Okay, so guys, this is the enzyme responsible for the polymerization of a nucleic acid, and this enzyme is also always synthesized. So this everything is actually synthesized inside of the G1 phase. Are you getting my point? So the G1 phase is nothing but the preparation for the S phase. Okay, in S phase DNA is copied. In M phase, actually cell divide its nucleus into the two. So the preparation for the division of a nucleus is happen inside of a G2 phase, and that's why the before S and the before M there is a one growth phase where the preparation or the cell is prepared for the next thing which is going to be happen inside of the cell division. Okay, so this is actually the theme behind the cell division. Let's see the one by one. We can see it not completely detailed today, but most of the thing we will discuss today here. Okay, so the mitosis, if you see, the mitosis is always start from the 46 46 chromosomes. So the 46 is for the human, guys. Okay, if you are talking about the honeybee, so it's a 32. If you are talking about the different animals, so the number of chromosome is a characteristics of animal, so the different animal had the different number of chromosomes. Okay, most of the time they can be shows the same number of chromosome, but if you see the complete overall structure and the overall the ATGC of animal, so you can understand the which type of animal it is. Okay, so this is not now very difficult to understand the animal from the chromosome and the DNA, because if you see the different type of methods of a DNA sequencing will help us to sequence the complete DNA into the 2 or 3 hour. Okay, so it will take only 2 to 3 hour. Okay, like your polymerase is now we are using it for the DNA sequencing. Okay, so the making of DNA by the polymerase will help us to understand the sequence of a DNA. So it is now not many I mean the very difficult to understand the a sequence of a DNA, and that sequence of DNA will definitely give us a knowledge about that particular animal, so which type of animal it is. Okay, so the let's say the 46 chromosome is a characteristic of a human, you can say. So the 46 chromosome cell is going to be enter into the division. So you can see here the cell is moving toward the both side, so the DNA material is also moving to both sides. So the one side is here, one side is here. Okay, so finally you can see the 46 chromosome here and the 46 chromosome here. So the both cell having 46, actually the first cell having 46, that means here is the copy of a DNA will happen. Okay, otherwise the cell will get 23. Okay, but cell will have the 46. Cell कडे 46 पासून सुरुवात करून 46 ला दोन सेल एंड होतात, म्हणजे कुठेतरी कॉपी बनवली. So the first thing, the making of a copy is inside of the S phase. Okay, and the finally cell ends, I mean the division ends into the 46 chromosome. So the starting point is a 46 and the ending point is a 46. That's why it is called as equational division. So the started from the 46 and ends with the 46. So similar number, it will divide into the similar number, and that's why it is called as equational division. If there is any change in the number, I mean the if number of a chromosome reduces, so there is the reductional division. We are also going to focus today on that. Okay, so the stages of the mitosis. So the main stages of mitosis is here. So the mitosis, the first stage is interphase. Okay, so the interphase म्हणजे काय, guys? So the interphase is nothing but the G1 phase plus S phase plus G2 phase. So these three phase togetherly called as a interphase. Okay, so interphase म्हणजे नवीन शोध लावायचा नाही. interphase म्हणजे आपल्याला माहित आहे. interphase म्हणजे काय मित्रांनो, तर काही कोणाचे प्रश्न असतील तर विचारा बरं का. तर G1 प्लस S प्लस G2 या तिघांना एकत्रितपणे इंटरफेस म्हणायचं, बाकी काहीला डोकं लावायचं नाही ते. ठीक आहे. सो द DNA is copied here. So the one cell which is having the pair of a chromosome, we can write it like a 2n. Okay, so that is 2n here. So the next phase, that is the interphase G2 is now end. So this is nothing but your M phase. Okay, and this is the cytokinesis, the division of the cytoplasm. Cytokinesis is the division of cytoplasm, and the division of nucleus is called as karyokinesis. Okay, so this M phase is nothing but the karyokinesis. Okay, so guys, in a prophase, if you see the chromosomes are present in the pair. So why there is a pair? If you see, this is the mother cell. So the first cell which is going to enter into the division is called as mother cell. Okay, so this is the mother cell having a single copy of a chromosome here and the single copy of a chromosome here. But if you see inside of the cell, So we have the two copies of a chromosome here and the two copies of a chromosome here. So the both cells, I mean having the two copies of a chromosome. So the actually copy is made up in this phase. Okay. So the out of two copies, if you see the chromosomes are very close to the equator of a cell, so they are held at the equator of a cell, and the equator, so this is the middle part, and that's why the name of a phase is a metaphase. Meta stands for middle. Okay, so the meta stands for middle. If you see now, the chromosomes are pulled toward the both end. Okay, so here is a one centriole, here is a one centriole which is pulling. Okay, दोघं पण काय करायला लागले, ओढायला लागले. Chromosome half half distribute झाले. So this phase is called as anaphase, where the chromosomes are moving toward the opposite pole, and the last one is a telophase, where you can see the division, I mean the starting of a division of a cytoplasm. And then one thing which is not shown very clear here, that is the telophase always shows the reappearance of a nucleus. Okay, so the nucleus is reappeared, and at the before the end of a telophase, nucleolus is also reappeared, and the finally you can see the cytoplasm is also divided, the one for each cell, and the finally we have the cell that is the similar like a mother cell having a single copy of a chromosome. These two single copies of a chromosome, these are nothing but the homologous chromosome. Homologous means one is from mom and one is from dad. Okay, so these are the homologous chromosome, and if you see the copies, so these two chromosomes is called as sister chromosome. So the both from one parent. Okay, either the two chromosome of a mother or the two chromosome of a father is considered as the sister chromatic. Chalo, let's see the next one. So this is the prophase. Here the what is happening inside of a prophase. So the first thing, the condensation of a chromatin material. You can see the chromosome is going to be compact. Okay, so the deappearance of the nucleus. So at the stage, I mean the late stage of a prophase, the nuclear membrane is going to be disappear. Okay, so the migration of centrioles is also happen. So the centrioles are moving toward the opposite pole in this phase. Appearance of mitotic division will happen. Okay, so the mitotic apparatus, sorry, not a mitotic division, but the mitotic apparatus, this is nothing but the spindle fiber. Okay, so this is the spindle fiber which is coming from the centrioles. So these threads which is moving toward the chromosome is appeared in this stage. Okay, so the from both chromosomes, these threads are coming moving toward the chromosomes. Okay, to find to hold the chromosomes, so they can pull them away from each other, and they will succeed into the segregation of a half chromosome to the new cell and half chromosome to the one second new cell. Okay, so this is the logic behind the mitotic spindles. Okay, so guys, we have one question. Sir, each cell in our body except of a germ cell do follow only this mechanism for cell division or what? Very good question. Okay, so the very good question. The confusion is always there for guys. Okay, so let's see. So the each and every cell of a body will enter into the mitosis. Okay, your which cell, what you talk about, the except of a germ cell. Okay, so the germ cell. So if you see the germ cell, so let's take an example of a spermatogenesis. Okay, so in the spermatogenesis, germ cell first divide into the two cell. Okay, so the chromosome number are very similar, 46. Okay, so this is nothing but the mitosis. So germ cell also enter into the mitosis. One cell going back to the first position to count the to keeping the count. Okay, and the second cell, we called it a spermatogonia, which will enter into the seminiferous tubule. Okay, so the lumen of a seminiferous tubule, or the adlumen of a seminiferous tubule. Okay, so here the primary spermatogonia is synthesized. Okay, so that is nothing but the growth between the spermatogonia and the primary spermatogonia, and this primary spermatogonia then enter into the meiosis. So till this stage, so this is the mitosis division. So you can say your germ cell is also enter into the mitosis. Okay, so the mitosis is compulsory for each and every cell which is present in our body. Okay, so the second thing, I mean the second question we have, and what about cell of other organism? Do they follow the exact same way for the division? Very good question once again. So the guys, the each and every cell that is eukaryotic. Okay, if it is going to enter into the cell division by the mitosis, so it will definite follow the same pathway. Okay, but few cells can enter into the amitosis. Okay, so in amitosis, so it is different. So here the actually the cell is like this. So the nuclear membrane is not going to disappear, but the nucleus is uh, divided into the two, and then cell will divide into the two. Okay, so this type of cell division is called as amitosis. Okay, so the sometime there is amitosis. Sometime if you see the yeast is also your eukaryotic cell, but it's a budding yeast. Okay, so the first thing is it is looks like this. So this bud is enter into the new cell like this, and finally you will have the two different cell from the. Okay, so this is also the mechanism of a cell division, but the mitosis is one of the most famous mechanism by which the cell can divide into the two. Okay, so if you see in mammals, in reptilia, in the fishes. Okay, so the after the porifera. Okay, so you can say that after the porifera, I mean this is the first phylum of animal. So all animalia group except of your the protista. So the protista had it's a one different type of a mechanism. They also follow the mechanism of the mitosis, but there is a few things like a fragmentation. Not a protista, but your fungus can fragment itself by the asexual reproduction. Okay, so the this type of things are there, but if you see the process of cell division, so it will definitely follow by your complete animalia. Okay, so the animalia, each and every cell will follow your the process of a mitosis, and the pathway for the mitosis is a very similar. So no one is going to bypass this type of a things. Okay, so it will result into the death of a cell if you bypass this. Okay, so we have one more question, sir. What is the exact difference between the mitosis and amitosis? Very good question. So yeah, actually the exact difference between the mitosis and amitosis. So the nuclear membrane is not going to uh, disappear into the mitosis. So the mitosis had it's a one pathway. It is different from the mitosis. So in the mitosis, the nuclear membrane is disappeared. Then spindle fiber comes together. They start moving toward the chromosomes. They will hold chromosome and pull them away from each other. But in the amitosis, everything which is going to be happen inside of a nucleus membrane is not going to disappear, and it will uh, cut into the two, and then cell cut into the two. Okay, so there is a difference of the appearance and disappearance of the nucleus. And the second thing, the pure mechanism which is absent in the mitosis. And okay, and the few mechanisms are also there which is absent in the mitosis, which is not a part of mitosis but the part of mitosis. Okay, आणखी काय असेल तर विचारू शकता तुम्ही. Okay, chalo. Next one. So in this stage, the mitotic apparatus is going to be appear. That means the what is mitotic apparatus? It is nothing but the spindle fiber. Let's see. The spindle fibers are made up of this mitotic. Let me clear it first. So this apparatus is nothing but the spindle fiber. Okay. So the spindle fiber also called as the microtubules. Okay, they are made up from the tubulin protein. ओके ट्युब्युलिन प्रोटीन सगळ्यांना माहिती आहे आपण सिलिया फ्लॅजेला पाहताना त्याच्यामध्ये पाहिला होता फ्लॅजेला जो आहे तो फ्लॅजेलिन प्रोटीन पासून बनतो अँड जो तुमचा सिलिया आहे तर त्यासाठी वेगळ्या प्रोटीन्स आहेत ओके तुमचं मायक्रोट्युब्युल जे आहे तर ते आपल्या मायक्रोट्युब्युल्स इज मेड अप ऑफ द ट्युब्युलिन प्रोटीन ओके सो दिस first phase second phase is a metaphase so why it is called as a meta because the meta word is the meaning of a meta word is a middle in the diagram you can see so here in the diagram the all chromosomes are very close to the middle part of a cell okay so they are at the equator okay so now we have the few names the labels here so guys so these are the mitotic spindles okay made up of the mitotic spindle which are made up from the tubulin protein okay so here we have few names so the first one is aster microtubule so aster microtubule which is moving opposite to the chromosome okay so the chromosome is not present here but they are moving toward the cell membrane and they hold this membrane so they can pull themselves toward the membrane or the pull membrane toward them but so it is not coming toward that so the automatically this assembly will move toward the membrane okay so it will result into the pulling to the chromosome and the chromosome gets separated from each other the one thing second thing the kinetochore microtubule so the kinetochore microtubule means any microtubule or any spindle fiber or the middle part of a chromosome okay so the middle part of a chromosome is called as centrosome okay and here is a few proteins which is looks like this okay so they are making the round on the this central part okay so let's see so these proteins are nothing but the kinetochores the structure is nothing but the kinetochore and actually the क्लियर करे आता okay chalo let's start finally we are again live okay so today guys I don't know why but there is a weak internet connection will not going to take us the continuous lecture okay so in the last point so where we was so I'm talking about the kinetochore let's see so this kinetochore microtubule so which microtubule is considered as a kinetochore microtubule okay so guys the microtubule which is connected to the kinetochore so what is meant by kinetochore let me draw first okay so for example this is your the chromosome okay so this is called as arm of chromosome it is again the upper arm of chromosome okay so this is the centrosome the middle part of a chromosome so depending upon the centrosome so sorry the here is the this is the central part it's not centrosome guys uh, this is the central part where the actually the structure is present okay so this the yellowish structure I am going to draw is called as kinetochore okay So this is the a proteinaceous structure. So when this microtubules come, they gripple themselves into the kinetochore like this. Okay, so they hook the kinetochore, and after the gripple with the kinetochore, they start pulling it toward the opposite pole. It will result into uh, the, the segregation of a chromosome. Okay, so the microtubule which is attached to the kinetochore is called as the kinetochore microtubule. ए येतोय का रे? आवाज येतोय का की बंद पडलाय? Okay, so this is called as the kinetochore microtubule, and the second microtubule which is coming from this end, and the one is coming from this end. So they just miss the chromosome and they move toward the opposite pole. Okay, so this microtubule is called as the interpolar microtubule. So there is always a motor protein between these two microtubules. So this motor protein actually are responsible for the pushing this microtubule into the opposite direction. So this is pushed toward this direction and this is pushed toward this direction. So this force is also added in the segregation of a chromosome, and they start the segregation, I mean the pulling of a chromosome from each other. So the one more thing, the chromosome that sorry, the microtubules which is called as aster, so they are moving toward the opposite pole of the chromosome. Chromosome is present in this side and they are moving toward the opposite pole. Okay, so here you can see this is the membrane of a cell. Okay, so this is the plasma membrane. So the this tubule attached to the plasma membrane, and they start the pulling this plasma membrane. But we know that the plasma membrane is held by the cytoskeleton. It is a hard structure. It is not going to change its shape very easily. So the plasma membrane is not going to pull toward the or move toward the microtubule, but in the opposite, microtubule is start moving toward the plasma membrane. Okay, because of this pulling force, they are start pull toward the plasma membrane, and automatically this force is applied to this point also, and it will result into the pulling of a chromosome, and the chromosome, your chromosome is looks like this. Okay, so when the chromosome is pulled toward this portion, okay, so it will looks like this. Okay, so the middle part is moving toward the, okay, middle part is moving toward the opposite pole. So this chromosome is looks like this. Okay, so this is actually the chromosomes are segregated, and the main role which is playing in the segregation of the chromosome is your centriole. Okay, so this centrioles and the spindle poles are the main player which is going to uh, pulling, I mean the main role, playing a main role in the pooling of the chromosomes. Okay, so in this structure you can see the anaphase. The chromosomes are looks like a banana. Here you can see. Okay, the chromosomes are looks like a banana. They are looks like this. Okay, so the group of a banana, we know. So you will have the question in examination, guys. So in which of the following phase the chromosomes are looks like a banana? So in that condition, you have to write that the anaphase where the chromosomes are looks like a banana. Okay. So in this structure, the very right side will show you that the kinetochore microtubule, or you can say the motor proteins. Okay, so in that motor proteins and the kinetochore microtubules, so one more thing is there, that is the tubulin subunit. At this point you can see, okay, so the tubulin subunit lost due to the depolarization. So what happened actually, guys? So if you see, the structure is made up of the tubulin protein. So this structure is made up of the tubulin protein like this. Okay, for example, these are the tubulin protein which is get attached to the centriole. Okay, so here is your centriole. Okay, so whatever it is a structure, but I'm going to write it like this. So the what happened? So the centrioles are start pulling it. That means they are again going to be shortened. So how? So the this centriole is going to attach to the kinetochore, and this is going to be a move. So what happened? So this patch is already open, and this is attached to this, means the chromosome is moving toward the uh, this one. So the in the next condition, so this is going to be a remove from the microtubule, and the chromosome is moving toward this part. Okay, so after the next stage, it is also going to remove, and the chromosome is moving to the next part. So then after this, the chromosome is again moving to the next part. Okay, so this chromosome is again moving to the next part. So this is actually the process, the cyclic process, where the one by one tubulin protein is going to be a depolarization or polymerize, and this depolymerization of the tubulin protein result into the pulling of the chromosome toward the end of the apparatus. Okay, so the spindle fiber moving toward the centriole, we can say. Okay, so let's say the telophase, it is one of the last phase of the young phase. Okay, so where the nuclear membrane and the nucleolus both are going to be reappear, because the now the chromosomes are completely separated, and we have the two nucleus in the single division, sorry, in the single cell. But here is the furo, which is start moving toward the each other, and it will result into the complete two cells. Okay, so the contractile ring, that is nothing but the furo here. You can say the end, the telophase ends when the contracting rings start forming. Okay, so let's see the where contracting ring now moving toward the each other, and they will join them together, and it will enter into the, I mean, it will aid into the two different cells. Okay, so the two cells are there. Okay, and these two cells uh, separate from each other, so that is nothing but the separation or the division of cytoplasm. Okay, and the division of a cytoplasm is called as the cytokinesis. So the cytokinesis is nothing but the distribution of organelles also. Distribution of cytoplasm, organelles, okay, and etc etc things are distributed in a cytoplasm, uh, because before the cytokinesis, if you see, the here is only one thing, that is the karyokinesis. In the karyokinesis, the division of nucleus or the separation of nucleus happen. Okay, so the division of nucleus is the first thing, and the division of cytoplasm is the second thing in the cell division. Okay, so the meiosis and the mitosis. So you can see the two things together here. So the in the mitosis, it is very easy. So the one cell is going to copy its DNA, and after copying the DNA, cell is going to distribute this DNA into the two, and ends into the same number of chromosomes. So for example, this is started from the 2n, it will enter into the 2n and the 2n, so 2 cell of a 2n. But here if you see, the first thing very similar, these two stages are very similar. The DNA is replicated here, and the replicated DNA is now not going to directly separated from each other, but here is a mixing of DNA. So in this phase, you can see the mixing of DNA is happen. Okay, and this mixing is called as the chiasmata formation or the crossing over. Crossing over. So this is the unique property of your meiosis, and this meiosis will happen. I mean the reason behind the variation in animals. Okay, so the when we called it about the genetic, so we know the 23 chromosomes are coming from the mother and 23 chromosomes are coming from the father. But if you see, we are not exactly seems like a mother and we are not exactly the looks like a father. So why it is happening? Why there is a difference in the appearance of our body and the father's body, or the appearance of the mother's body and our body? So this is only because of the variation which is happening inside of the meiosis. Okay, so the crossing over in the meiosis is a very important property. It is actually responsible for the making of a difference between the two brother and the two sisters. Okay, or you can say the in between the brother and sister. If you see in the brother and the sister, so the 23 chromosome which is coming from the same mother and 23 chromosome which is coming from the same father, still they are very different from each other because of the only this meiosis process. Okay, and nothing but the meiosis. Okay, so let's say what happened, the mixing of a chromosome is happen and then they are going to divide into the two. So after the one division, the meiosis one is completed, and then again cell do not enter into the this S phase, or the do not enter into the interphase. So here is the interphase. We know what is interphase. So the interphase is a G1 plus S plus G2. Okay, so in the second division, that is meiosis 2, and the first division meiosis 1, so there is a one phase. It should be a interphase, but the interphase is absent into the meiosis 1 and meiosis 2, and that's why the cell which is the result of a meiosis 1 again enter into the division, and it will result into the again two cells. So the one cell, that is the mother cell, is divided into the four cell of the four haploid cell. So in the meiosis what happens, the one cell is divided into the four haploid cell. Okay, so the one diploid cell enters into the four haploid cell, and in the mitosis one diploid cell enter into the two diploid cell. Okay, so the number of cells are more in the meiosis. So if you see, the property of meiosis 1 is the crossing over, and the property of meiosis 2 is the formation of a four cell. So in the first division here you can see the chromosomes are going to be separate in this division. Okay, so these separated chromosomes are nothing but the father's chromosomes are moving toward the one cell, and the mother's chromosome is moving toward the one cell. You can see here the maternal and paternal we already written here. So this is the red color. So paternal means coming from the dad, and the brown color which is coming from the mom. Okay, so these two chromosomes, one is coming from the dad and one is coming from the mom. It's called as, so for example, this is a dad's chromosome and the mom's chromosome. This is called as homologous chromosome. So these homologous chromosomes are separated in the first division. So I'm going to write here the homologous chromosome. So already written here, and homologous chromosome is going to be separate. Homologous chromosome, and in the second division here the chromosomes which is going to be separate from each other, nothing but the sister chromosome. So what is meant by sister chromosome? For example, I am taking this mother's chromosome separately, and this chromosome is getting, I mean, the copied into the two, and this chromosome is also copied. So we have the one copy of the chromosome of a mom and the chromosome of a dad. So these two chromosomes which is a copied, so it's called as nothing but the sister chromosome, and this is also called as the sister chromosome. So the sister chromosome is nothing but the pair of a chromosome coming from the single parent. Okay, or you can say the copy of a chromosome is called as sister chromosome. Okay, so this is actually the process of meiosis. Let's see how it is happen. So the meiosis does two things. So if you see, the we are the sexually reproduced organism. A the 46 chromosome cell is get fused with the 46 chromosome, so it will result into the 92 chromosome. So in the next generation, this will going to cross with the 92 and it will enter into the 184. So each and every division will increase, I mean each and every sexual reproduction will increase the number of chromosome. So it is very necessary to maintain the number of chromosome. So in that condition, so the meiosis is a very important thing, where the 46 chromosome cell is converted into the 23 chromosome gamete. Okay, and the 23 chromosome gamete. Okay, so whatever the male and the female gamete is. Okay, so they are coming together and they form the again the characteristic of a human, that is the 46 chromosome. So to maintain the count of a chromosome in the sexual reproduction, it is very necessary to have the meiosis in a body. Okay, so the second thing, the one meiosis is going to convert the one cell into the four haploid cell, that is the four gamete. So the making of more number of gamete required the meiosis process. The second thing, that is the meiosis going to be the result into the independent assortment and the crossing over. So in the crossing over, what happened? The chromosome from the mother and the chromosome from the father, they are going to be mixed with each other. So the for example, this red color chromosome from the mother and which is going to be attached to the father's chromosome, and the blue color chromosome of a father is going to half of the part is or not a half, but the part is going to attach to the mother's chromosome. So this is called as a crossing over. The mixing of a two chromosome which result into the diversity in the animals. Okay, so this is called as a crossing over. Let's see the next one. So the meiosis, in the meiosis process, you can see the first of all, the chromosome is copied into the two. Okay, so the chromosome, parental chromosome, is copied into the two. This copied chromosome is divided into the first division, which sphere is getting a split. So the mother's chromosome is moving toward the one set and the father's set is moving toward the second cell. Okay, so in the second division, so this single chromatid is going to be separate into the single cell, and that's why result into the four cell, the four haploid, and here the two haploid. Okay, so the meiosis one, it separates the homologous chromosome, the very important topic. Okay, so the homologous chromosome. So this first division of a meiosis is also called as heterotrophic division, because the parental cell divides into the two haploid cell. So it is started from the 46 chromosome and ends into the 23 chromosome. So the started from the 2n cell and end into the n cell, and that's why it is called as heterotrophic, because it is not end into the same type of a cell. Okay, and the interphase is completed now in the meiosis. Before the meiosis 1, each chromosome is replicated. It results into the two genetically identical sister chromatids which remain attached at the centromeres. Okay, so the prophase 1. So in the prophase 1, it is a crucial phase of meiosis. During this phase, the chromosomes are get separated, and after the synapses or the crossing over, and this the chromosome which is coming together is called as a tetrad. So I will show you the diagram of a tetrad. So the crossing over is the exchange of a segment during the synapses. That is the part of a meiosis 1. It takes some more time, and the phase is the peculiarity of meiosis. Okay, so this phase is actually the star phase of a meiosis, which is the peculiarity of a meiosis. Let's see how it is going to be happen inside of a cell. So guys, this is your prophase 1. Okay, so in the prophase 1, the chromosomes are moving toward the the prophase 1, we know the chromosomes are moving toward the centrioles are moving toward the opposite pole. So the one is moving toward this and one is moving toward the this pole, and the chromosomes are here between the two centrioles. Okay, so they are at the opposite pole now. So in a 180-degree they arrange themselves. So here the prophase 1 is again divided into the 5 phases. Okay, so these 5 phases can be the mug or can be keep in mind like this. So the first phase is a leptotin. So from the leptotin, let's take LA. So lazy paddy. Okay. So this lazy paddy word will help you to keep in mind this the stages in the sequence. Okay, with the sequence. So the first LE stands for the leptotin. So in the left protein, if you see, the chromosomes are condensed and they are coming very close to each other. Okay, the second ZY stands for the zygote. Okay, so the zygote is the formation of a bivalents. So the chromosomes are coming closer and they form the attachment between the two chromosomes. So for example, this is a one chromosome having a copy, this is second chromosome having a copy. uh, so this is second chromosome having a copy. So when they comes closer, they form the protein structure which attach them to each other. So this protein structure which attach this chromosome to each other is called as synaptonemal complex. सिनेप्टोनेमल कॉम्प्लेक्स म्हणजे काय मित्रांनो, तर सिनेप्टोनेमल कॉम्प्लेक्स म्हणजे प्रोटीन स्ट्रक्चर जे दोन क्रोमोझोमला एकत्र धरून ठेवतंय. ओके, का धरून ठेवतं बघा, तर थर्ड मध्ये पीए जो आहे, सो दिस पीए स्टँड्स फॉर द पॅकेजिंग. Okay, so this PA stands for the packaging phase. So in the packaging phase, chromosomes are now completely attached to each other because of the synaptonemal complex. And in the DI phase, so it's a diplotin. So in this phase, the chromosomes are moving from the each other. I mean, moving away from the each other. And in that phase, what happened? So this synaptonemal complex is moving toward the pole of the chromosome, and that's why the chromosomes are now attached like this. Okay, so this one chromosome from the mother and the second chromosome from the father, they are going to see like this. Okay, so the if you see, this portion is completely get merged into the each other, and finally they detach from the each other like this. Okay, so this chromosome is detached like this. Okay, and the father's chromosome is detached like this. So the mother's chromosome is having the piece of a father's chromosome now. Okay, so this bluish color, and the father's chromosome is having the piece of the mother's chromosome now, this reddish color. Okay, so this is nothing but the crossing over, the mixing of a chromosome, or the what we call it, the sharing of a chromosome or the exchange of a chromosome is called as the crossing over, which is happening inside of the packaging. So please keep in mind that the condensation and the coming closer of a chromosome is happen inside of the leptotin. So in the zygote, synaptonemal complex can be appeared in the packaging. So this crossing over will start in the diplotin. So the crossing over dissolve, I mean ends, and the only chiasma you can see. Okay, so the dissolution of synaptonemal complex is started into the diplotin, and in the dikinesis, so dy, this is the last phase. So in the dikinesis, the nuclear membrane start dissolving and the chromosomes are separated after exchange of the material. Okay, so this is happen inside of the prophase 1. So the if you see the prophase 1, so the crossing over is happen like this. So this is the chiasma formation in the first phase. So the chromosomes are breaking, and the joining of a chromosome is happen at the point of a crossing, and after the crossing over the recombinant chromosomes, you can see the piece of a red chromosome is shared to the blue chromosome, and the piece of a blue chromosome is shared to the red chromosome. So you can see the red is a dad's chromosome, and this is the mom chromosome. So the chromosome from the two parents are going to mix with each other, and it will result into the recombination. Okay, so that recombination is actual the process which is responsible for the what we call it, the variation into the organisms. Okay, so the same parent will give the birth for the two children. If you see, the both children are not similar. Okay, so if there is the twins, they are also showing few differences. It is only because of the uh, the synaptonymal complex and the crossing over. Okay, so this is the meiosis process which is responsible for the variation in the organism in the sexual reproduction. Okay, so the next one is a metaphase. We know the PMAT. Okay, so the PMAT is the actually the sequence of a phase which is going to be happens. The first is a prophase, second is a metaphase, third is a anaphase, and last one is a telophase. Okay, so this is also common for the mitosis and it is also common for the meiosis. Okay, so the mitosis and the meiosis, for the both processes, these four steps are very common. And now we are in the metaphase 1, that means the meiosis, the metaphase of the meiosis 1. Okay, so we are here in the meiosis 1, and the prophase is we completed with the prophase is sorry, the prophase is classify into the 5 phases, that is we already completed in this leptotin, zygotin, packaging, diplotin, dikinesis. These are the nothing but the 5 phases which comes under the prophase 1, and now we are in the metaphase 1. We know the metaphase stands for the chromosomes are at the middle or the equator of the cell, and that's why we call it a metaphase. Okay, so this is the metaphase where the chromosomes are coming at the equator and start segregating from each other. So anaphase where the chromosomes are looks like a banana and they are moving toward the opposite phase. We all know the characteristics of anaphase. We already discussed into the mitosis. So after the prophase 1, if you see the process of mitosis, it is very similar. Sorry, the process of meiosis is very similar like a mitosis. Okay, so the only prophase 1 where the chromosomes are getting mixed. So after that, the metaphase, anaphase, and telophase. These three phases are very similar like a mitosis. So the things which is coming under the anaphase is very similar. Telophase very similar like the process of telophase in a mitosis. Okay, so the cell is going to be a divide, and here the cytokinesis 1, the two daughter cells are going to be formed. Okay, so the one cell is now divided into the two daughter cells. So this is a 2n cell and this is the young cell. Okay, so young cell having all chromosome from mom. Okay, and it is having all chromosome from dad. सगळे पप्पाकडले क्रोमोझोम एका बाजूला मम्मीकडले क्रोमोझोम एका बाजूला केले. So you can say it's a segregation of homologous chromosome. Okay, so in the meiosis 1, so this is nothing but the meiosis 1 process. Okay, so after that cell is going to enter into the meiosis 2. Okay, so this is a meiosis 2 where the one cell is going to separate into the two. Okay, so we know that the one chromosome is already copied into the two, and that's why the one is moving toward the one cell and second is moving toward the second cell. So it is a division of a sister chromatid. Sister chromosome is going to be a divide, and that's why we have the 4n cell at the end of the meiosis 2. Let's see the meiosis 2. So the meiosis 2 is the second meiotic division and also called as homotypic because it is started from the n cell and ends into the n cell. That's why it is called as the homotypic. But if you see the meiosis 1, so meiosis 1 is started from the 2n cell and ends into the n cell, so it is called as heterotypic division. Heterotypic division. Okay, so the heterotypic means the different thing. 2n into the n is a heterotypic and n into the n is a homotypic because the number of chromosome in the first stage and the last stage very similar. Okay, so the two haploid cell forms during the meiosis 1 further divided into the 4 haploid cell without entering into the interphase. Okay, so the interphase is not going to be happen between the two meiosis 1 and the meiosis 2. Otherwise, in the interphase, we know that there is a formation of a chromosome. It will again form the chromosome, and then again number of a chromosome will increase. So that's why, to avoid the more number of chromosome, the cells are going to be skip that interphase and immediately enter into the meiosis 2. Okay, so the 4 haploid cell is a characteristics of a meiosis 2. These cells are the similar to their parent with respect to the chromosome number during the meiosis 2, and so called as homotypic division. I have already explained here. So if you see that this meiosis 2 is a very similar like a mitosis. How? Let's see. So in the meiosis 2, this phase is like a mitosis. Okay, so sometime you will ask, you will have the question in examination, which of the following phase is a very similar like a mitosis. So please click on the meiosis 2. Meiosis 2 is a similar like a mitosis. Okay, how? Let's see. So what happened in the meiosis 2, the prophase 2, where the nuclear membrane is going to be a disappear at the last of the prophase 2, and the chromosomes are going to be sorry, the spindle fibers are going to be appeared, and the metaphase 2, so the chromosomes are at the midpoint of the cell, or at the equator of a cell. In the anaphase 2, the chromosomes are looks like a banana, and they are moving toward the opposite pole. In the telophase 2, so the everything which is the nuclear membrane and the nucleolus, all the things which is disappeared in the prophase, so they are going to be appeared in the telophase. And finally cytokinesis 2, where the cell is going to be divided into the 4. So in these stages, so there is no mixing of a chromosome, no synaptonemal complex is there, no crossing over is there, no synapses is there, and because of that it is looks like very similar like a mitosis, and that's why this phase is called as a mitosis phase. Okay, not called as a mitosis, but called as a very similar like a mitosis. So the meiosis 2 is a similar like a mitosis, and it is called as the homotypic division. Okay, meiosis 1 is called as heterotypic division. Okay, so overall meiosis process, if you see, it is started from the 2n cell. It is started from the 2n cell and ends into the 4 cell of the n. Okay, so the n and n. So the finally 46 chromosome cell is enter into the convert itself into the 23 chromosome cell, and that's why the number of chromosome is going to be reduced, and that's why the meiosis is called as a reduction division. Reduction division. Why reduction division? Because the number of chromosome is going to be the less at the final point. Okay, so let's see guys. So this all about the cell cycle and the cell division. Okay, so the final difference, let's see the final difference I'm going to discuss here. So anything that is cell want to be a repair or reproduce or copy, so these things are done by the mitosis. Okay, so in the cell, where is the meiosis actually happening? So meiosis only result into the formation of a gamete, so it is required for the sexual reproduction only. Otherwise growth, repair, reproduction, replacement of a cell. Okay, so this everything which is happening inside of a body is because of the mitosis. Okay, so guys, please keep in mind that. Okay, so the mitosis is for the replacement, repair, everything which is the present inside of a body is a replace, repair, reproduce, copy, or the growth of that particular the thing is because of the mitosis. Okay, and this meiosis is only responsible for the sexual reproduction. Okay, so do not get confused with the meiosis and mitosis. Meiosis never used to increase the number of cell in the body or for the growth of a body. It is a very very specialized division for the formation of a gamete. Okay, लक्षात येतंय. आणि जे काय बाकीच्या गोष्टी आहेत, so I also explain about the death of a cell. So why apoptosis is unnecessary and what is meant by apoptosis, and what is meant by necrosis, what is meant by PCD, programmed cell death, and why they are required. So I already explained into the mitosis. So the mitosis is a special lecture you will have on this channel. So if you want to see that lecture, so guys, please see in the description you will have the list of my lecture, a playlist of my lecture. You can click on that. So guys, one more thing I want to told you that I want to say you about that. In the description of this video there is a one the feedback form. If you want something from us, you can add that thing into the feedback form. So I request you, please fill that feedback form. So it is about everything, so how your educators are, if you like it, so please write, I mean the type there like, if you don't like it, so please give the reason. So these things are absent into the lecture, so we require that thing. Whatever you require you can fill there into the feedback form, so I will definitely give you the that things. Okay, so guys, this is the Telegram channel, so it will definitely I will explain this thing. So it is for the MCQs practice and the notes. ओके, MCQ सोडवायला एवढी मजा येते याच्यावरती की त्या पोल फॉर्मॅट मध्ये असतात, मग तुम्ही ते क्लिक केलं की ते फटाके वगैरे वाजवतं जर बरोबर आलात तर, आणि नाही आलात तर मग तुम्हाला माहिती आहे ते काय काय दाखवतं, ते गेल्यावर कळेल तुम्हाला व्हायब्रेशन्स वगैरे सगळ्या गोष्टी. दिस इज द युट्यूब चॅनल. ओके, पहिल्यांदा त्याला सबस्क्राईब आणि बेल आयकॉन दाबून ठेवा, आणि ज्यावेळेस पण अनअकॅडमीला सबस्क्राईब करताय तर हा कोड आहे मित्रांनो, हा कोड वापरायला विसरायचं नाही. It will definitely reduce your fees. Okay, so it is for the discount in a fees. Capital Abhijit, त्याला सबस्क्रिप्शन कोड म्हणतात. ठीक आहे, so thank you very much. Thank you very much guys. If you like my lecture, please click on a like and see you today 7 p.m. So today at 7 p.m. we will have the revision on the developmental biology. Okay, that is the embryogenesis. Okay, so the embryogenesis we will discuss on the 7 at the 7 p.m. today. So thank you very much. See you at 7 p.m. today.